Okay, let's talk about the sodium potassium ATPase pump. So I'm going to kind of throw this back on you. A little active recall. All right, so first off, we actually just kind of talked about it. Write out the biochemical equation for ATP hydrolysis that is performed by the sodium potassium ATPase pump. Right, so first, we can ask multiple questions about this pump. What is the biochemical equation for ATP hydrolysis? Start with the molecule of ATP. What else? Is it reactant? H2O. Okay. We're going to have some enzyme hydrolyze this. I'm going to actually use this specific one. The sodium potassium ATPase pump is an enzyme that hydrolyzes this reaction. All right. So those are the reactants. What are the products of ATP hydrolysis? ADP, adenosine diphosphate, plus an inorganic phosphate, a loose one, plus energy. Okay, so the sodium potassium ATPase pump, now I drew a cell. Let's put this in context. It hydrolyzes that ATP and converts that energy, that stored potential energy, and it converts it into kinetic energy. What does it move? How many in which direction? Talk to a neighbor. It's moving sodium. How many in which direction? 3 out. And potassium in which direction? 2 in. All right, so why do we need a molecule of ATP or energy from ATP to move sodium out of the cell and potassium into the cell? Why? Okay, because we're moving sodium against its concentration gradient, potassium against its concentration gradient. So if we can draw in 4 ions. All right, drawing the concentration gradients for sodium, potassium, calcium, and chloride. So we already mentioned sodium is higher outside the cell than inside the cell. Potassium is higher inside the cell than outside the cell. So again, we're moving both of them against their concentration gradient. All right, then is calcium typically higher outside or inside the cell? Typically higher outside the cell. Shown by the larger symbols. All right, and then what about chloride? Typically higher outside the cell than inside the cell. Okay, so again, we need to utilize directly a molecule of ATP to move sodium against its concentration gradient, potassium against its concentration gradient. What do we call that process by which we use energy and ATP to move a substance or an ion against its concentration gradient? And you can be a little little bit more specific. This is an example of primary active transport. All right, so ATP is directly used. In primary active transport, ATP is directly used to move things against its concentration gradient. All right, so then how does primary active transport differ from secondary active transport? Okay, secondary takes advantage of the energy used in primary active transport. So typically we use sodium in a co-transporter. Sodium will, the large driving force will go from outside the cell to inside the cell because of the concentration gradient, and then it's so powerful we can pull either amino acids or glucose into the cell against their concentration gradient. So secondary active transport takes advantage typically of the sodium concentration gradient to pull things into the cell against their concentration gradient. Okay, then let's draw in some ion channels. So I'm going to draw an ion channel in for each one of these ions, a transmembrane protein. Okay, this sodium channel opens. Which direction will sodium flow under normal conditions? So we call this sodium influx. Again, the natural tendency for sodium, without energy, the natural tendency, sodium will always move down its concentration gradient from high concentration outside the cell to inside the cell. What about calcium? What's its natural tendency if that channel opens? Into the cell, influx. Okay, chloride, influx. And what about potassium? Efflux. So it will leave the cell, say through a potassium leak channel. Natural tendency. Okay, what do we call that process by which things are naturally moving down their concentration gradient from high to low, but they need a membrane protein in order to do so? Facilitated diffusion. So all of this are examples of facilitated diffusion. Diffusion, things always move from high to low concentration, but because these ions, the membrane is impermeable to these ions, we need a helper protein that has access to outside and inside the cell to allow that to happen. Okay, so primary active transport, secondary active transport, facilitated diffusion. What are substances that can move across the plasma membrane via simple diffusion? Gases, so oxygen, carbon dioxide, nitric oxide are going to be the three big ones we talked about. And then what other substances? Steroid hormones. Hormones with a lipid backbone. Again, this is a phospholipid bilayer plasma membrane. Lipids love lipids. Right? So hormones with a lipid backbone can freely move via simple diffusion from high to low concentration. Okay. Yeah. We hit a lot there. What other questions? There's a whole lot going on. Yep. as we're starting out here at this part of the semester, the inside of the cell is always going to be negatively charged, have a negative resting membrane potential as compared to the outside. Okay. Two of the main reasons shown here, we have tons of potassium leak channels in our cells, so if they're always open, there's no gate, so potassium constantly leaks out. Okay, so if you have a positive ion leaving this space, it's going to make outside more positive, inside more negative. In our sodium potassium ATPase pump, if we have an imbalance, 3 positive charges leaving, only 2 coming back in, it makes the outside more positive than inside. And then the third reason why excitable cells have a negative resting membrane potential, there tend to just be more negatively charged anions like bicarbonate inside the cell than outside the cell. So those are the 3 reasons why there's a negative resting membrane. When we get into the nervous system, we'll start talking about depolarization and hyperpolarization, but we're not there. But does that clarify your question? Answer your question? Okay. Yeah.
What's your study was on the online peer that's all those topics from like 1 through 4. So this is the week study guide. Is that going to help or is there?
And I just use the prep guides that I made for you to help focus. Each chapter has its own prep guide. That is your study guide. They're on Brightspace. Each week's folder has a prep guide, chapter 1 prep guide, or week 1 prep guide. So use those to help focus and narrow your study. But there's more in the chapters than I could ever cover. So please focus, use those to focus on being efficient with your study. Other questions? Yeah. Sure.
I know that's like a very broad topic.
Yeah, no, you can. And I'll use a visual. All right, so I'm going to kind of use my drawing here. All right, so again, your cells are constantly getting different signals to either turn on genes or turn off. We're going to focus more on turning it on. So that signal, again, on the nuclei of all of your cells, you have all these genes that are located in double-stranded DNA. That's the blueprint for all of our proteins. All right? We do not want to mess with the blueprint. We do not want to mess that up. That can lead to disease or disorder. So you get some signal, they write down the instructions for that protein, so they transcribe it or write it down into messenger RNA. That messenger RNA is single-stranded. It's small enough to move through the nuclear pores into the cytosol or cytoplasm. The ribosomes are the machinery that then dock to that mRNA and then can also have a transfer RNA that also dock to it, because in that mRNA there's a series of nucleotides in a very specific sequence that then, that's just the written instructions. We need to actually translate it or make that protein or start making that protein. All right, so we take those instructions and then we create a polypeptide. Polypeptides are too immature and they're in the wrong shape to be fully functional. So then we need to process them. After they're translated in the rough endoplasmic reticulum, they might string together a series of polypeptides or modify that polypeptide into a more complete protein, but it's not folded yet. And so then it must go to the Golgi apparatus to be folded correctly, and then it is functional. So structure dictates function, so it has to be in the correct shape for it to function correctly. That was very fast. Follow-up questions on that? Again, all these proteins we've been talking about, sodium, potassium, ATPase pump, voltage sodium channels, potassium leak channels, aquaporin channels, are all going to have a different gene or multiple different genes and then fold differently. They have different folding because they're different amino acid sequence, therefore different functions, different structures, different functions. How's that? Yes? Other questions on transcription, translation? Okay, there's 5 minutes left. Something that we have not talked a lot about, because it was from week 1, which, again, there's going to be a dozen questions on language of anatomy from chapter 1, all right? Quick review, all right? We made up a song, all right? All right, what do we got here? Spell? Acromial? Teller? Toes or fingers are both digital, all right? So it's going to be cephalic, acromial, patellar, digital, and then orbital, otic, oral, okay, talk to a neighbor, find these on your person. A quick review.
Yeah, so there's vertebrae. The cervical is not cervix. It's different. The shape of the face. Wait, I remember a question. You're right. Oh, yeah. Digital is your femur is your thigh. This is your butt. Lumbar is your lower back. This is your hand, right? Manual. Oh, that's the way I remember. Manual hand. Venus. Mental is your foot, no?
Yeah.
Am I wrong? I thought I got what's my name? Oh, your chin, sorry.
And focus more on the ions. We've been talking about in terms of elements, what makes a protein, what makes a lipid, what makes a carbohydrate. So focus on the ones more that we're going to touch.
Right, you don't talk about this thing.
Chemical bonds, yeah, covalent versus ionic versus like bonds, where they're located, and more applied to why it's important physiology.
Yeah, you said it. I just
All right,
Neo is the post
3 minutes left. Any clarifications? Anything? Yeah.
Wait, that's I forgot. This real new.
What's that? Coronial? Coronial? No. Coronial, there's a synonym for coronial, the fibular region. So everyone touch your fibular region or your coronial region. All right, it's on the lateral aspect of the lower limb. All right, so coronial versus perineal. Perineal is the perineum, the space between your genitalia and your penis. Perineal region. Any other clarifications?